Hello, hi, good evening. So good evening, Om sir. So it's really happy to see you on a Saturday evening. So thank you for joining me on this weekend lecture. So we have been talking about the journey of cell, and cell is definitely the superhero of biology. So by now we are done with almost 3 lectures, and you would have almost concluded that cell is definitely the superhero of biology. So thank you for joining me in this journey. But don't please forget to like, share and subscribe. So please support us. It's a new channel and we are looking for a broader reach. So make sure you like, share and subscribe. Good evening, Harini. Okay, so we are in lecture 4 today. So I believe we'll complete the chapter today. So it is the end of cell, the superhero of biology. So the next class will be on Tuesday. So wait for the interesting session. So I'm not disclosing what the session is about, but it's definitely an interesting session. So wait for my thumbnail, wait for my update. I will definitely not disappoint you again. Okay. Okay, so before actually going to the lecture, so make sure you download this app, eCareerPoint app, and you can find my PPTs here. So you can join the eCareerPoint family. So you will get to know a lot of things. Fine. So good morning, Anil. Sorry, good evening. So it's good evening, not good afternoon. So I guess all of you are still sleepy after lunch. So after 4, it's good evening. So good evening, Anil. Okay. So we were so long talking about the cell. So what did we actually talk about? So a quick recap of all your 3 lectures. So we are talking about why the cell makes you living and what differentiates between you and non-living. And we've also been talking about why the cell is unique time and again. Right? So all the 3 lectures I've been talking to you about the uniqueness of the cell. So it is completely independent. So if you leave a cell alone, it can survive. Right? So it's completely independent. Then we were talking about the cell theory. Yes, good evening. So we have been talking about the cell theory. So we have 3 players in cell theory. The first one was Clarence Swan, who gave us cell theory, but they failed to mention how the cells arise. So then you have another player who's called as Rudolph. So Rudolph here gave us the modified cell theory that we are reading today. So he said cells arise from pre-existing cells, and that was one groundbreaking discovery of the cell theory. Okay. So overview of the cell. So we have been talking about the cytoplasm and plasma membrane. Your cell basically contains cytoplasm and plasma membrane. So what is the difference between cytoplasm and plasma membrane? So cytoplasm is a fluid matrix present in all your cells, whereas plasma membrane is the delimiting layer of your cell. Okay. So then we have been talking about nucleus. So nucleus is present in both prokaryotes and eukaryotic cell as well. But what is the major difference? So your nucleus is not well defined in case of your prokaryotes, but it's well defined, pretty well organized in case of your eukaryotes, right? So that's what we've been talking about, the overview of cell, and we spoke about the uniqueness of plant cells and uniqueness of your prokaryotic cells. So what is unique in prokaryotes is your ribosomes. So prokaryotes do not contain any other organelles except your non-membrane bound ribosomes, because ribosomes are important for your translation or your protein synthesis machine, right? The next one is your animal cell. So animal cell again has a non-membrane bound organelle, and it is called as a centrosome. Good afternoon. So that is what we've been talking about. Then it's good evening again. So prokaryotic cell. So we have been talking about prokaryotic cell. In prokaryotic cell, we spoke about the cell envelope modification. So cell envelope was divided into glycocalyx, plasma membrane, glycocalyx, cell membrane, and the plasma cell wall and the plasma membrane, right? So these were the 3 modifications of the cell membrane of your prokaryotes. So that is what we have been talking in the prokaryotic cell. So that was followed by the inclusion bodies. We read about the rhizosomes, we read about the slime layer and the tough capsule. That is a modification of your glycocalyx, right? So going on further, we were still in this eukaryotic world. So eukaryotic world seems so endless. So we have been talking about time and again about the eukaryotic world, and it seems to be never ending, because your eukaryotic cell has a lot of organelles. So many organelles that each organelle has a unique function, and hence it allows the harmonious interplay between the cells. Okay. So now going forward and revising your last class lecture. So we were here, we were talking about your endomembrane system. So your functions are coordinated. So when I say system, it means it has 3 or 4 organelles whose functions are coordinated. Okay, it includes endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. So why these 4 come under endomembrane system is that their functions are interlinked. So from proteins, your endoplasmic reticulum synthesizes proteins, and from proteins they go into Golgi complex where your proteins are packaged, Okay, so once they are packaged, one packaged vesicle becomes your lysosomes, and your lysosomes contain hydrolytic enzymes which helps in digestion, intracellular and extracellular digestion. Fine, and that is followed by vacuoles. Okay, so vacuoles are more predominant in a plant cell and they occupy 90% of your cell volume. Fine, so this is your endomembrane system. So next we were talking about your mitochondria. So mitochondria is your double membrane structure and it is also known as the powerhouse of your cell. So it has an inner membrane and an outer membrane. So it has your inner membrane space. Let's quickly go to the diagram because since it is a recap. So this is the structure of your mitochondria. It has an outer membrane, it has an inner membrane, and there's a space between the two membranes which is called as the inner membrane space. Then it has numerous foldings on the inner membrane and these foldings are called as cristae. So in case of endoplasmic reticulum and Golgi body also you have foldings and these foldings are called as cystae, but in case of mitochondria these foldings are called as cristae. Fine, is it understood? So this is about mitochondria, and the fluid matrix present on the inner surface of your mitochondria is called as matrix. So remember, mitochondria starts with M. Thus the inner fluid substance, the dense substance that is present is called matrix. So again that starts with M. So that is how you remember. Fine. So going on further and talking about the structure of chloroplast. So it is a double membrane bound. Okay. It's a space limited by inner membrane. So there the space in case of mitochondria was matrix. In case of chloroplast, it's stroma. Okay. So you have flattened membranes called thylakoids. Then you have thylakoids arranged in stacks or piles, that is called grana. So let me quickly show you the structure and explain. So this is the structure of chloroplast. You have an outer membrane and you have an inner membrane. So the space between outer membrane and inner membrane is your inner membrane space. Okay. Fine. So inner fluid matrix here is called as stroma in case of chloroplast. So you have flattened membranes which is called as thylakoids, and thylakoids contain your chloroplast. When you have stacks of thylakoids, it's called as granum. Okay. So your grana are connected with the help of lamella, and the space inside your thylakoid is called as lumen. So this is where we were talking about in the last lecture, that is on Wednesday. So we were talking about the structure of mitochondria, structure of chloroplast, and we continued with the next slide today. So that is, we are going to talk about cilia and flagella. So it's not that cilia and flagella are only present in the prokaryotic world. So cilia and flagella are also present in the eukaryotic world. So we have been discussing this time and again, right? So your body and my body has ciliated epithelial cells in case of your respiratory tract. So it helps in purifying the air which enters your body from the atmosphere. So that is also one function of your cilia. So today we're going to look at the structure of how your cilia and flagella are designed, how intricately they are designed to carry out their function. So it is one of the beautiful architecture in your entire cell. So it is called as a 9+2 arrangement. So you would have come across a lot of questions which has this 9+2 arrangement. So it is a beautiful architecture. So that is your cilia and flagella. So basically your cilia and flagella are air-like outgrowths of your plasma membrane. So if you go back to my prokaryotic world, so there was a slide where I showed you all the three structures. One is flagella, one is cilia, and the other one is, it was flagella, cilia, pili and fimbriae, right? So these three structures. So they were just hair-like outgrowths from your cell membrane. So one was little long, the other was pretty small, right? So that was the difference. So they are basically hair-like outgrowths of your cell membrane. So cilia are small structures which work like oars. So movement of either cells or either they help in the movement of the surrounding fluid. So they work like oars. So when you are traveling in a boat, so you sail the boat, right? So either you move the boat against the water or you just maintain your flow in water, right? So that is the same function of your cilia here. So flagella, they are longer and they are responsible for the cell movement. So I've been always telling you, so right now your body and my body has a lot of bacteria floating inside. So they float with the help of flagella. Okay. Flagellum and cilia, so singular ones are called as flagellum and cilium. So plural is called as flagella and cilia. Okay, so they are covered with your plasma membrane. They emerge from centriole-like structures which is called as your basal bodies. Fine. So this is your cilia and this is your flagella. So your flagella basically helps in locomotion. So they are pretty long structures. So they are longer compared to your cilia. So that is the difference between cilia and flagella. Okay. So structure of how your cilia and flagella looks like. So that's what we are going to talk about. So the core of your cilia and flagella, from which your basal body arises, is called as axoneme. Fine. So let me go into the structure and explain you all these terms. So this is how it looks like. Okay. So this is beautiful, right? It's a very good architecture that you can see. So this is called as the 9+2 arrangement. So where does this 9+2 come from? So if you look into the periphery, you have these beautiful structures, right? So these beautiful structures are called as the radially arranged peripheral microtubules. So these are the radially arranged peripheral microtubules. Okay. So they are in doublets. So each microtubule has 2. So it's called as a doublet. So there are 9 of them. If you have patience, start counting 1, 2, 3, 4, 5, 6. So there are 9 of them. And in the center, you have a separate pair, right? So it is called as the 9+2 arrangement. And the center one is enclosed with this red color. So this red color is called as the sheath. Fine. So these two are joined with the help of bridges. And this sheath, it is joined with the peripheral microtubules at a particular point, right? So each of them touches your sheath. So that point where it touches the sheath is called as your radial spokes. Fine. And each of the adjacent microtubules, they are also connected. So nothing is in isolation. So everything is connected with each other. So they are connected with the help of linkers. Fine. Is this understood? So now looking into the statements here. So that's what I have explained. So the core is called as axoneme. Fine. So there are 9 doublets of radially arranged peripheral microtubules. Plus you have a pair of centrally arranged microtubules. Thus it is called as the 9+2 arrangement. So remember, 9+2 arrangement is very common for your cilium and flagellum. Fine. So you have bridges. So they are joined with the help of bridges and linkers. So you have a central sheath here. So this is the design of your cilium and flagellum. So from your from your cilia and flagella, you have what is called as a basal body formation. Okay. Fine. So centrosomes and centrioles. So these are the next categories. So these terms are pretty confusing. So you tend to confuse these two terms, centrosome and centrioles. So there's also another term which people tend to confuse, that is called a centromere. So in biology, these 3 terms are pretty confusing. So these 3 terms are pretty confusing. So now can you tell me the difference between the 3? So your centrosome contains centrioles and they are involved in pulling of chromosomes during the cell division. But centromere is when you draw a chromosome, this area is called as a centromere. Fine. Is this understood? So your centrosome is a non-membrane bound organelle that contains 2 cylindrical structures that is called as centrioles. Fine. Your centromere is the one which is present in the middle of a chromosome. Okay. So centrosome is an organelle containing 2 cylindrical structures and that is called as centrioles. Surrounded by amorphous pericentriolar materials. So quickly let me show you the picture again. So this is how your centriole is. So there are 2 cylindrical structures. They contribute to what is called as your centrosome. So centromere is in case of chromosomes. So centrosome consists of 2 cylindrical structures and they are called as your centrioles. Fine. Is this understood? Can you see these 2 cylindrical structures? So here also again you have 9 microtubules. So 7, 8, 9. So you have 9 microtubules here. But if you look closely, so they are in triplets. So you have 3 of them making 1 microtubule. So they are in triplets. Can you see? It has 3. So they are called as your peripheral microtubules and the center portion is called the hub. So this portion is called the hub. It is made up of proteins. The central portion is connected to the periphery with the help of spokes again. Is this understood? So they form your basal bodies and this is your centriole and your centrosome. Fine. So now are you clear with these 3 terms, centrosome, centriole and your centromere? So in case of cell division, I am sure all of you would have seen images like this. So initially your chromosome will align at a particular place and on 2 poles you will have some features which will pull your chromosomes apart. So the one that is pulling your chromosome apart is your centrosome and your centrosome is made up of centrioles. So if you look properly, you have 2 cylindrical structures here. They are your centrioles. Fine. Is this understood? Okay. So nucleus. So that is your next organelle. So we are going to talk about nucleus. So nucleus is the last organelle that we are left with. So talking about nucleus, we'll talk about how your DNA is organized, how your DNA is precisely packed, accurately packed inside your cell, and what is chromosome. So can we study the chromosome? So these are the last topics for today. So with that, your cell will be done. So we're almost done with the superhero of biology, and today is just the climax of the entire story. Okay. So talking about nucleus, so nucleus is the brain of your cell. So just like you have the CPU as the brain of the computer, so your nucleus is the brain of the cell because it contains the genetic information. So for time and again, I've been talking about the central dogma of life. So central dogma of life says that information is copied from the DNA to the mRNA, and from there it is translated to proteins, right? So basically, your nucleus gives command, right? So that is why your nucleus is called the brain of the cell. So let's see. Leeuwenhoek first observed nucleus in the RBC of a fish, okay? And also to make one point clear, your RBCs, that is mature mammalian RBCs, do not have a nucleus. They are basically enucleated. For that point, they do not have any membrane-bound organelles. So they do not have nucleus. They do not have endoplasmic reticulum. They do not have mitochondria. They do not have chloroplast. Fine? Is this understood? So that is one exception, because in every aspect you have an exception, right? So in biology, we'll talk about a lot of exceptions. So one such exception here in your first chapter is your RBC, mature mammalian RBC. It does not have a nucleus. It does not have membrane-bound organelles, okay? But your immature RBC has a nucleus, but as it matures, it removes its nucleus and it becomes enucleated. Fine? So Robert Brown in 1831, he identified nucleus, okay? So basically your nucleus is again double membrane. So can you quickly tell me what are the double membrane organelles in your cell? So recently also, just a few minutes back also, I was revising the double membrane organ, your mitochondria, your chloroplast, and now your nucleus is also double membrane. So you have a question in NEET asking which of the following organelles are single membrane, and another question stating find the organelle with double membrane. So make sure when you're studying, when you're preparing this chapter, you pay focus on the structure as to how they are designed, whether they have double membrane or whether they have a single membrane. Fine? Is this understood? So double membrane bound dense protoplasmic body controls all cellular metabolism. Yes, so all your cellular metabolism, the instruction, the command comes from your nucleus, right? So it encloses the genetic information of the cell. So that is why it is very, very important. So nucleus is absent in mature phloem and sieve tube elements and mature RBCs of mammals. So your phloem, it's a part of your vascular system in plants, your phloem and xylem. So your phloem also does not have RBC, as well as your mature RBC in mammals. So your RBC and my RBC do not have nucleus, but the immature RBCs do have a nucleus. Fine, is this understood? Okay. So now actually talking about the structure of nucleus. So during the interphase stage of nucleus, so all of you know your cell has to divide. So we all start from a single organism, right? So we all start from zygote. So zygote is single cell, and you cannot see a cell in a naked eye. But today, you and I have such huge structures, so we are multicellular, right? So how do we become that multicellular organism? So what makes us look multicellular? It's basically one thing. Your single cell divides and divides and divides to produce faithful copies of itself. Thus, today we arise as multicellular organisms. So when does this division happen? So there are two types of division. First is the division of your nucleus. That is, your genetic material is equally distributed. Fine. So such a division is called as karyokinesis. Karyo means nucleus. Kinesis means division. Okay. And then cytokinesis. Cyto is cytoplasm. Kinesis is again division. So first your nuclear material will divide, followed by the division of your cytoplasm. Okay, so is this understood? So now again I'm talking about the double membrane structure of your nucleus. So there's a space between the inner membrane and the outer membrane, and that is called as a perinuclear space. Fine, is this understood? So the outer membrane is continuous with your endoplasmic reticulum. Hi Priyanka. So it has two membranes, outer membrane and inner membrane. So in between both the membranes, you have a perinuclear space. Fine. So the outer membrane is continuous with your endoplasmic reticulum. And since it is continuous with your endoplasmic reticulum, so your endoplasmic reticulum has ribosomes attached to them, which forms your rough endoplasmic reticulum, right? So your ribosomes are attached to the outer membrane as well. Then you have your nuclear membrane. So your nuclear membrane is perforated by minute nuclear pores. So this is about your inner membrane that I am talking about. So you have pores on the membrane, so which allows the transport of substances into the nucleus and out of the nucleus into the cytoplasm. Fine. So each pore, that is, we are talking about nuclear pores here. So each pore is guarded by the octagonal discoid structure of the nucleoplasmin protein. So that is called as annulus or bleb. So understand, so you have nucleus. So your nucleus is something like this. So I have the picture in the next figure. So your nuclear membrane is something like this. So you have pores in between, which allows the transport of proteins and RNAs into the cell and out of the cell into the cytoplasm. Fine. So each pore is guarded by an octagonal discoid structure of a particular protein. So this setup is called as annulus or bleb. Fine. Okay. So annulus plus your nuclear pore forms your nuclear pore complex. Fine. So is this understood? Okay. So now look into the structure of your nucleus. So it is again a double membrane structure. So outer membrane is continuous with your endoplasmic reticulum. So your endoplasmic reticulum has ribosomes attached to it. Right? So you have nuclear pore here. So all these are your nuclear pores. So the inner side of your nucleus is called as your nucleoplasm. So this entire region is called as nucleoplasm, and nucleoplasm consists of nucleolus and your chromatin. So chromatin is your genetic material. So in the next slide we'll talk about how your chromosomes, how your DNA is packed inside the cell. So they are packed as chromatins inside the cell. So basically inside the nucleus. Okay, so apart from chromatin, your nucleoplasm also has nucleolus. So nucleolus is the ribosome factory of your cell. So when we were talking about ribosomes, we studied that ribosomes arise from the nucleolus. So ribosomes are the protein factory of the cell, and that ribosomes arise from your nucleolus. So nucleolus is present in high concentration in actively protein synthesizing cell. So this was once your NEET question. Okay, fine. So now going forward, so the nuclear pore, what is the main function of your nuclear pore? So your nuclear pore mainly regulates proteins moving into the nucleus and out of the nucleus. So basically your RNA and proteins. So when you understand the process of central dogma, you will understand replication and transcription happens inside the nucleus. But once your mRNA is produced at the end of transcription, your mRNA has to move into the cytoplasm for the process of translation. So you need to transport the mRNA out of the nucleus into the cytoplasm. Okay, fine. So that is one of the role of your nuclear pore. So the inner side of the inner nuclear membrane is lined by nuclear lamina. So that is called as nuclear lamina, and it is made up of laminin proteins. So everywhere you have a lot of proteins. So here the nuclear lamina is made up of your laminin proteins. Fine. So the core complex provides main channel. So it forms a barrier between your nucleoplasm and the cytoplasm. So we are talking about your nuclear pore complex, that is your annulus plus your nuclear pore, that is your nuclear pore complex. Fine. Okay. So what are the functions of your nucleus basically? So ribosome formation. So basically it's your nucleolus that helps in the ribosomal formation. Then it enholds or it encloses your genetic information. So it contains your DNA. Then transmission of genetic information. Then you have cell division happening, because I told you about karyokinesis, where your nucleus first divides to produce faithful copies of itself. That is, it ensures that the genetic material is being passed on. Fine. Then control of metabolism. Yes, your nucleus actually controls which protein has to be produced and at what time. Suppose if your body does not have one process happening, it will turn off the genes of that process so that energy is not wasted. Fine. So it controls the process of metabolism. So next one, how DNA is packed inside your cell. So this is very important. So your DNA is very huge, right? So you have 10 power 14 cells, and each cell has a DNA of 2.2 meters. So then quickly calculate how much DNA you have. So it is pretty big, but your cell is pretty small. So how can you accommodate such a huge DNA into your cell? So that is one of the brilliant way your cell manages to accommodate DNA. So there are various ways of packaging by which your DNA is accommodated inside the cell. Okay, so this is how it is arranged. So you know your DNA has double helical structure like this, right? So yesterday we were talking about, yesterday I was talking about the genetic world, where we were discussing the molecular basis of inheritance. So we were talking about the base pairs of DNA, the number of bases you have. So you basically have 6.6 into 10 to the power 9 base pairs. So this is for a haploid individual. Okay, it is so long. So you have so many base pairs. But how this is being compressed, how this is being condensed. So that is a beautiful process which you and I did not design, but it was designed by nature, and we are studying that. So you all know this double helical structure of DNA, right? So these double helical DNA, they are negatively charged because they have a phosphate group. So your DNA is made up of nucleotides. So nucleotides have 3 components. So the first component is your nitrogenous base, second one is your sugar, and third one is your phosphate. So when your nitrogenous base, that is your purines and pyrimidines, bind with a sugar, it's called as a nucleoside. And when a nucleoside binds with a phosphate, it becomes a nucleotide. So because of the presence of phosphate group, your DNA is negative. So here comes another player called as histone proteins. Yes, Priyanka, you're right. So histone proteins come into the picture. They come in aid to compact the DNA. So your histone proteins are a group of related proteins which are positively charged. So now your negatively charged and your positively charged proteins will come together. So these round structures are your histones. So they are called as histone octamers. So you basically have H1, H2A, H2B, H3 and H4. So these are the 5 histones that you have. So except histone 1, all these 4 histones, they form what is called as a histone octamer, and that is this round molecule. Okay, so they form that round molecule. So around that round molecule, you have your DNA wound around it. So it's basically like this. So let me write it here. So you have this histones. So your DNA is wound around your histone. It's basically like a string on a bead structure. Okay. So when your histone is wound around the DNA, it is called as nucleosome. Fine. Repeating structures of nucleosome is called as chromatin. So you have repeating structures of nucleosome, it is called as chromatin, and further condensation forms chromatin loops, right? So this is chromatin loops, and that further condenses to form condensed chromatin loops, and finally you have what is called chromosomes. So it will remain in your nucleus as chromatin loops at the time of cell division, at the time of M phase. It will compact, it will condense to form what is called chromosomes, and inside your chromosomes you have your genetic material and a particular sequence on your genetic material that codes for your protein is called as gene. Fine. So you understand how beautifully, how amazingly your cell accommodates DNA, though it's so huge, right? So this is how your DNA is packed. So I guess no doubts in this, so I'm going forward. So next we'll talk about chromosomes. So what are chromosomes? So the term chromosomes was first proposed by Walder. So basically the scientist's name are not important, but sometimes you cannot always depend on your luck. So it's okay. So just put a reminder in your brain. Okay, so you have read about a scientist called Walder. So put that in your brain at the back. If at all you get a question, and if that strikes you, then luck plays a role. As such, they have not asked about scientists till now. But remember, it is not bad. So chromosome was proposed, the term chromosome was proposed by Walder. So your chromosomes are best studied at metaphase. So what is this metaphase stage? So during cell division, you have 4 phases. So cell division happens in 2 phases. One is called as interphase and the other one is called as the mitotic phase. So interphase is the preparatory phase where your cell prepares itself for the division, and M phase stands for your mitotic phase. And under this mitotic phase, you have 4 subphases. It is called prophase, metaphase, anaphase and telophase. So let me write it fully for all those who have just joined. Prophase, metaphase, anaphase, telophase. Okay. So during prophase, your chromatin will condense to form chromosomes. During metaphase, so suppose if this is your cell. So during metaphase, your chromosomes will align together at a particular equator like this. So when it aligns together at a particular equator, it is easy for you to study the morphology of chromosomes. So morphology is the external appearance of your chromosome. So in the next coming slides, I have a picture of all your 23 set of chromosomes, and you'll be amazed to see not all the 23 chromosomes look exactly like each other. Some chromosomes are short, some are long. Okay, so they are not identical. So you can study the morphology of chromosomes at the metaphase. So that is what this point says. Fine, is this understood? So 2n is the number of chromosomes in diploid cell. So humans are diploid. So we have one copy, 1n coming from your mother and 1n coming from your father. Okay. So n is the number of chromosomes in haploid cell. So basically they are your gametes. So your x and y are the gametes. So x has haploid, y has gap haploid. So when your zygote is formed, you become a diploid individual. Fine. Okay, so you have your cell, so you have chromosome. So chromosome contains your DNA. Is this understood? So this is how chromosome, DNA, chromatin, everything comes into picture. So I hope there is no confusion. So that's why I discussed how the chromosomes are packed. Is this understood? Okay. So going forward, so we'll talk about karyotyping. So what is karyotyping? So is this a new term to you or it's an old term? So for all those who do not know what is karyotyping, it is a study of external morphology of chromosomes. So just now I was telling you all your 23 pairs of chromosomes do not look identical. Some look long, some look short. Okay, so not all 23 are the same. So to study the external morphology of chromosomes, we have a term that is called as karyotyping. Fine. So how do you study this external morphology of chromosome? So your chromosome is like this, right? So this is called as arms. So these are called as arms. So also known as chromatids. So based on the length of your arms, based on the number of chromosome that is present, based on the size, based on the position. So where is your chromosome present? So based on the position of your centromere. So sometimes your chromosome can be like this as well. Okay. Sometimes it can be in the center. Sometimes it can be like this also. So based on the position of your centromere, the length of arm, the secondary constriction. So this is your chromosome. So the place where the chromatids are held together is called a centromere, and this is known as primary constriction. So apart from this, if you have a secondary constriction, it is called as satellite. So these are called as constrictions, that is there's a small bend in your chromosome. Okay. Yes, ideogram is a diagrammatic representation of chromosome. So basically you do karyotyping in case, so these days in hospitals you have genetic counseling. So to ensure that the kids are not born with any genetic defect, so that is one of the preventive measures that is coming up. So what do? You study the karyotype of the fetus. Okay, so before it's being born, so in a particular trimester you study the karyotype of the chromosome. So it is basically a computer based software. So you get a final representation, I have it here, and that is called as ideogram. Fine. So based on these characteristics, you can study the external morphology of chromosomes, and that is called as karyotyping. Fine. So ideogram is the diagrammatic representation of karyotype, and this is how your ideogram looks like. So it's basically a computer software, and it is not easy to do this. It's a very tedious process. You have to take out a particular tissue of your fetus. You have to stain them. It's not easy. It's easier said than it is being done. So everything is tedious because inside your body they are stable. But once it comes out of your body, it is really tough to maintain the conditions inside your body. You have to mimic the conditions that are present inside your body. Okay. So this is the chromosomes. Fine. So look at here. All the chromosomes are not identical. Each chromosome is different, right? So look at the shape of this chromosome. Look at the shape of this chromosome. So why do you have two pairs? So this is a diploid organism. You have one copy of chromosome, one from your father and one from your mother. So it is diploid. So there's a defect here. So if you look into the X chromosome, so it has XXY. So it has two X chromosomes. So such a type of defect is called as Klinefelter syndrome. So we'll talk about the genetic disorders. There we'll discuss more in detail about the Klinefelter syndrome. But as of now, understand, so why do we study such the external morphology is to know where the defect is. In case of transgender people, so that is a chromosomal disorder. They have one X chromosome extra like this. Okay, so only when you study the karyotyping, you will know where the mistake is. Sometimes a portion of this will be deleted, so you will have a defect. So that is why you study what is called as karyotyping. So you have karyotyping now done for almost all diseases, all genetic diseases. So you can just Google it up if you're very interested. So do it for Down syndrome, do it for Turner syndrome. So all are basically chromosomal abnormalities. So we do study the karyotyping. But these days, genetic counseling is coming up at a high speed. So where for each fetus, for each kid before birth, that is in the particular trimester, karyotyping is available. For that, you study the pedigree of the family. Some disorders are sex linked, right? So based on that, you understand the family history. Then you come to a conclusion whether the child is susceptible to this disorder or not. Then you go on to do karyotyping, because it's pretty costly. It is not feasible always, and it is not an easy process as well. Fine? Okay? Fine. So the last topic for today is your microbodies. So with microbodies, we'll be ending your chapter, the cell, the superhero of biology. So what are microbodies? They are small bodies. So we call them as membrane-bound minute vesicles. So they are called as microbodies. So that contain various enzymes. So they're present in both plants and animals. Fine. So the first one is your peroxisome. So peroxisomes are more predominant in plant cells. So peroxisomes contain enzymes. So basically they catalyze your redox reactions, that is reduction oxidation reactions. So they're basically minute membrane-bound vesicles which contains enzymes for a particular type of reaction. So that is called as peroxisomes. Then it is sperosomes. So sperosomes are lysosomes, are equivalent to ribosomes present in your animal cells. So they basically contain lipids or fats. Okay. Then glyoxysomes. So glyoxysomes contain fats. So they basically present in immature plants. Before the plants can actually perform photosynthesis, these supply nutrients to the plants. So these are microbodies present. They are not exactly organelles, but you call them microbodies. The last one is transosomes. So transosomes are vesicles present in the ovary of large birds. Okay, so they have a particular function there. So that is also one of the microbody present in your eukaryotic world. So with this, we have successfully completed all the organelles of your prokaryotic and your eukaryotic world, though your prokaryotic world has only a non-membrane bound organelle that is called as ribosomes. Fine, is this understood? So this is how your microbodies look like. So they are a minute single membrane bound organelle. So they contain a crystalline core where they contain enzymes for a particular reaction. Okay, fine. So let me quickly recapitulate. In case of eukaryotic cells, we have spoken about a lot of organelles. If you remember, first we started off with the plasma membrane, where we spoke about the fluid mosaic model. So the fluid mosaic model gave way to the types of transport. So we spoke about the types of transport. Then we spoke about the cell wall, where we spoke about the middle lamella, the function of the cell wall, right? So then we entered into what is called as the endomembrane system. So endomembrane system consists of your endoplasmic reticulum. It consists of your Golgi body. It consists of your lysosomes and vacuoles, right? So after endomembrane system, we were talking about your mitochondria. Then we were talking about your chloroplast, right? And today we entered into what is your cilia and flagella, the structure of your cilia and flagella. And then we continued to talk about what are centrosomes and centrioles. Finally, we spoke about your nucleus, which was followed by your chromosomes, and finally microbodies. So this is in short. So when you are revising this chapter, so this chapter has a very less weightage. So the weightage of this is 2% in your NEET exam. So when you are doing your final revision, so quickly write down whatever you have studied. So that is the best way to recollect. So write down eukaryotes and write down all the organelles that you have studied. Now come back to plasma membrane. Just recite the points that you know. So it is made up of phospholipid bilayer, so that you are thorough with the concept. So when you are talking about mitochondria, quickly make a rough figure of mitochondria, so that when you have diagram based questions, you do not go wrong. So that is how you revise this chapter. Fine. So it's a pretty simple chapter. Since we have just started on this journey on YouTube, so I thought I will go with a very easy chapter. So we are done with Cell, the Unit of Life today. So it took us 1 week to talk about the superhero of biology. But I hope I didn't disappoint you with this chapter. So the next class will be on Tuesday for Standard 11. So Monday, that is on that is Monday at 4 o'clock. I'll be talking about again genetics, that is class 12. Okay, but Tuesday session I'm not revealing. It's a special session. So be curious, so be interested, be very anxious, because it's a short and a very interesting session. So stay updated and stay tuned to know what is that session. Okay, so wait for my thumbnail. So until then, have a very happy weekend. So if you really like my session, share it, subscribe it, and share. Let your friends know that there's something going on like this. Okay, so have a very good weekend. So until then, it's me signing off. So see you on Monday exactly at 4 p.m. Thank you.